therapy. Hypersensitivity is an inappropriate immune response that results in host damage, and two major types are antibody-mediated and cell-mediated hypersensitivity. Hypersensitivity diseases are categorized according to antigens and immune mechanisms that produce disease, and they can take between minutes, like hay fever, to hours, like an allergy to penicillin, or to days, up to 2 days, like poison ivy response. Immediate hypersensitivity, an example is allergies, which are antibody-mediated immediate hypersensitivity, and they're caused by the release of vasoactive products from mast cells, which act on blood vessels. And mast cells are a type of leukocyte, which play a role in inflammation. And these reactions occur within minutes after the exposure to the antigen. They can be mild or life-threatening, like anaphylaxis, and about 20% of the population suffer from allergies, which is hay fever. And then there's delayed type hypersensitivity and cell-mediated hypersensitivity, which would be T cells, respond to using tissue damage like hives, and this is due to inflammatory response. Symptoms can appear several hours or even days following secondary exposure, such as poison ivy blisters. And typical antigens include microbes, self-antigens, and chemicals that covalently bind to skin, creating new antigens, which would be contact dermatitis. Here's a picture of the raised red areas after contact with allergens from immediate hypersensitivity. And then these are poison ivy blisters from delayed type hypersensitivity. Autoimmune diseases occur when T and B cells are activated to produce immune reactions against self-proteins. This results in host tissue damage, and some diseases are caused by autoantibodies, which interact with self-antigens. And examples of this would be allergic encephalitis and type 1 diabetes. Superantigens are proteins capable of eliciting a strong response because they activate more T cells than a normal immune response. They are produced by many viruses and bacteria that interact with T cell receptors. And superantigen activated T cells may produce systemic diseases characterized by systemic inflammatory response. Examples of superantigens are rheumatic fever, which would last a few days and is a complication of Streptococcus pyogenes infection, arthritis, Kawasaki syndrome, atopic dermatitis, and some forms of psoriasis. Immunodeficiency is active adaptive immunity, which is critical for infectious disease resistance. Animals have deficiency in B cells and are prone to bacterial infections, and animals that have deficiency in T cells are prone to viral infections and cancers. Severe combined immune deficiency syndrome is a serious inherited deficiency of both B and T cells, and patients will live a restricted life, limiting their exposure to any pathogen. And then acquired immunodeficiency syndrome is caused by HIV infection that progresses and kills CD4 plus T cells. And these patients are prone to opportunistic infections and cancer since they are deficient in T cell help. And then here is the concept map so far. Okay.
Okay. I'm Ellie Carroll, and I'm gonna take over after Lillian. So we have the vaccinations are basically just deliberate exposures to antigens which trigger an adaptive immune response intended to protect an individual against future infection by that same pathogen. The immunogen used to induce this artificial active immunity is called a vaccine. Active immunity is memory cells, when memory cells are generated. And then in the table on the next slide, only examples of vaccines for major diseases like anthrax, cholera, diphtheria, and meningitis. And then that's that table. And then the nature of vaccines, it is recommended that infants be immunized against key infectious diseases as soon as possible so that their own active immunity can replace the temporary maternal passive immunity. It is well established that the introduction of effective vaccines into a population has reduced the incidence of formerly epidemic childhood diseases such as measles and mumps and has eliminated smallpox altogether. The nature of vaccines, immunization with live cells or a virus is usually more effective than with dead or inactivated material. Most agents used for immunization are either attenuated, which is weakened or inactivated pathogens, or inactivated forms of microbial products, such as toxins, also known as toxoid. Vaccines stop spread of infectious diseases, especially in kids, and the CDC sets guidelines in the US for those vaccines. Immunizations usually involve a series of secondary immunizations called boosters to produce a secondary response in a higher antibody titer. And those are just some of the recommendations for infants and children's for the vaccines. Synthetic and genetically engineered vaccines. Synthetic vaccines are the product of genetically engineered antigenic components. Conjugate vaccines attach smaller antigenic protein to larger carrier protein to improve immune response. Attaching a polysaccharide to a toxoid improves immune response to polysaccharides. And pneumococcal vaccines are made this way. Nucleic acid vaccines, they're vaccines which the antigens of interest are usually synthesized by the animal. DNA vaccines are bacterial plasmids that contain cloned DNA that encodes the antigen of interest. RNA vaccine contain bRNA sequence encoding agent and antigen. Use a novel vaccine delivery method, not injected into the animal. Target proteins are cloned into the plasmid vectors and injected intramuscularly. The DNA, RNA is taken up by host cells and proteins are expressed, meaning transcribed and translated. The host then mounts an immune response to the protein. Field in clinical medicine that seeks to harness cells and of other components of the immune system to fight or prevent diseases, including various forms of cancer. Key immunotherapeutic approaches currently in development to combat cancer are anti-cancer vaccination, use of checkpoint inhibitors, adoptive T cell transfer. Anti-cancer vaccines can be either prophylactic, which means preventative, or therapeutic. Prophylactic vaccines target the oncogenic, also known as cancer-causing pathogens, which means no disease equals no cancer. Human papillomavirus, also known as HPV, can cause cervical and other forms of cancer. And hepatitis B virus can cause liver cancer. HPV in the US is high risk. HPVs cause 3% of all cancers in women and 2% of cancers in all men. And transformation in eukarya is a process by which virus infection can lead to the cell becoming cancerous. And therapeutic anti-cancer vaccines are designed to stimulate the body's natural immune response to those cancer cells.
Okay, I'll show the concept map again. Okay, there it is.
I'm Mallory LaHost, and I'm taking over after Ellie Carroll. So, checkpoint inhibitors' goal is to prevent neutralization of cytotoxic T cells and enhance tumor cytal effector activity, keep T cells going. The most successful checkpoint inhibitors are monoclonal antibody MAB based drugs. Marketed as Keytruda treats metastatic melanoma and prevents it, which is skin cancer, from coming back after surgery. 10 years survival is 22%. Targets PD-1 program death-1 on T cells, or its ligand PD-L1 on cancer cells in solid tumors, and blocks inhibition of the anti-tumor immune response and keeps cancer cells from hiding. Side effects are autoimmune conditions. Not all patients respond, so primary T cells don't respond, and secondary new clones of tumor cells develop. Resistance is possible. Response linked to a specific gut microbiota. Adaptive T cell transfer. ACT focuses on harnessing the tumor targeting abilities of T cells. So two main approaches have been developed for ACT therapy: tumor infiltrating lymphocytes, which are TILs, tumor antigen specific T cell populations that can be isolated from resected tumor tissue and cultured, activated, and amplified in vitro, then infused back into patient, which is naturally present T cells. Chimeric antigen receptor, which is CAR T cells. T cells are designed to express modified TCR protein fusion derived chimeric antigen receptors, which are CARs, with enhanced antigen specificity targeting cancer cells, GMO T cells. And this is just the overview. Okay. Antibiotics are naturally occurring or synthetic antimicrobial compounds that are produced by fungi or bacteria. In order to be useful, antibiotics need to exhibit selective toxicity and target pathogens, but leave host cells and tissues unharmed. Antibiotics are classified by their mechanism of action, as each group targets different parts of bacterial anatomy or physiology. The susceptibility or sensitivity of microbes to different antibiotics varies greatly. Gram-positive and gram-negative bacteria vary in their sensitivity to antibiotics. Broad-spectrum antibiotics are effective against both groups of bacteria. Narrow spectrum only against certain groups. And this is the mechanisms of action for antibacterial agents. The cell wall as a drug target. So drugs that inhibit cell wall synthesis, peptidoglycan synthesis, include beta-lactam AB and others, so glycopeptide AB. Most of the over 100,000 metric tons of antibiotics used worldwide each year are beta-lactam antibiotics, such as penicillin and cephalosporin, effective against both gram-positives and gram-negatives, not effective against archaea and eukaryotes. Since eukaryotes do not have peptidoglycan cell wall, the side effects are non-existent unless you are allergic. The beta-lactam ring is found in penicillins and cephalosporins.
Okay, I'll show the concept map again. Is
Hi, I'm Ella Boudreau. This is just a little pie chart of the estimated 100,000 metric tons of antimicrobial agents that are manufactured per year. You can see that there's about 2 times as many agricultural than there are human. And nearly, oh, okay. There you can go back, or you can go forward. This is just another pie chart. It shows the frequency of antibiotic usage. It says that by far the B-lactam antibiotics are the most important and widely used class of antibiotics. Now looking at antimicrobial drugs and protein synthesis. Most of the antibiotics targeting protein synthesis will target translation by binding to the bacterial ribosome, both 30S and 50S subunits. Bacterial ribosomes are slightly different than eukaryotic, giving selective toxicity, which means that the eukaryotic host is not effective. Examples of the antibiotics that target protein synthesis includes aminoglycosides, and an example of that would be most things that start with strepto and gentamicin, tetracyclines, and macrolides, which is erythromycin antibiotics. Macrolides are a tunnel plug and they basically block new peptide exit tunnel. Resistance is the efflux of tetracycline or a protective protein for ribosomes. Here is a picture of staining of teeth from using tetracycline. Tetracycline binds calcium in the developing bones and teeth, which weakens them, and it can also stain your tooth enamel. Nucleic acid synthesis as a drug target, DNA replication and RNA synthesis, transcription. Quinolones are antibacterial compounds that interfere with DNA gyrase. An example would be blocking the supercoiling and packaging of DNA. Because DNA gyrase is found in all bacteria, quinolones are effective for treating both gram-positive and gram-negative bacterial infections. Resistance is a mutation in DNA gyrase or RNA polymerase gene that results in an altered protein, and it cannot be binded to. And they interfere with transcription. Other antibacterial drug targets, some examples are inhibitors of synthesis of major metabolites like folic acid. Some of these include sulfa drugs and resistance and development of alternative biochemical pathways. Analogs of growth factors, isoniazid is a growth analog effective only against Mycobacterium, and it interferes with the synthesis of mycolic acid. There's also an inhibitor of lipid biosynthesis, which is platinum targets fatty acid biosynthesis and a new structural class of antibiotic. It has a very broad spectrum and it's effective against MRSA and vancomycin-resistant enterococci, and it disrupts cellular membranes with polymyxins.
Is that okay? I can
I think, yeah.
share the new concept now. Go. Okay.
Okay, I'm Sophia Torrealba, and I'll be taking over after Ella. So this is antimicrobial drugs that target non-bacterial pathogens. Most antiviral drugs target host structures, resulting in toxicity. Most successful and commonly used antivirals are the nucleoside analogs. This blocks reverse transcriptase and production of viral DNA. It's also called nucleoside reverse transcriptase inhibitors. Non-nucleoside reverse transcriptase inhibitors bind directly to RT and inhibit reverse transcription. Fusion inhibitors prevent viral fusing with the host cell. Neuroaminidase inhibitors successfully limit influenza infection, which prevent viral enzyme from acting. Interferons are small proteins that prevent viral multiplication by stimulating antiviral proteins in unaffected cells. Drugs that target eukaryotic pathogens would be fungi, which pose special problems for therapy because they are eukaryotic. Much of the cellular machinery is the same as that of animals and humans. As a result, many antifungals are topicals, which is creams, and then about 40% of all hospital-acquired infections are fungal. So this is targets of some antifungal agents. And then for fungi, a few drugs target unique metabolic processes, such as cell wall synthesis, ergosterol inhibitors target the unique fungal plasma membrane component ergosterol. Allylamines, azoles, and drug bands such as grizofulvin. Cell wall inhibitors inhibit 1,3-B-D-glucan synthase that helps to make chitin and are used or chitin and are used to treat candida infections. Other target membranes or disrupt microtubule aggregation or target DNA synthesis. Like antibiotic resistance, antifungal resistance is on the rise. Okay. This is antimicrobial drug resistance and the acquired ability of a microorganism to resist the effects of a chemotherapeutic agent to which it is normally sensitive. There are at least 5 reasons that microorganisms are naturally resistant to certain antibiotics. An organism is impermeable to antibiotics. An organism can inactivate the antibiotic. An organism may modify the target of the antibiotic. An organism may develop a resistant biochemical pathway. And lastly, an organism may be able to pump out the antibiotic. Because antibiotic resistance genes are in nearly every population, physicians cannot use the same antibiotic for long. It takes 2 to 4 years to develop resistance. Any use of antibiotics select for resistance, increasing the number of resistant bacteria in any bacterial population. Bacterial resistance to almost all known natural antibiotic has been found, which in like deepest caves or overuse in cattle accelerates the spreading of the resistance. Some resistance forms arose recently. And this is the emergence of multi-drug resistance in gonorrhea.
Okay, I'll show the final concept map. Go. And then I think that's it.